Hi, my name is Dr. Shiva Ayyadurai. I'm a MIT-trained scientist. I hold 4 degrees from MIT, including my PhD in biological engineering. Originally, the word genetically modified organisms was referred to as GMOs, genetically modified organisms. Later on, any type of thing called a GE food or genetically engineered food also got the acronym GMOs. Right? So a GMO is a genetically engineered organism or a genetically engineered food. That's what a GMO is. The products that come out of genetic engineering or GMO would never occur in nature. So for example, you're taking a genome or a piece of a gene and you're putting it into the gene of a tomato because maybe the flounder can withstand cold, and we identify the gene in there, which is a gene that can withstand cold weather, and we want to be able to grow hardier tomatoes, so you're transferring the gene from the flounder into the gene of the tomato. Now obviously a tomato and a flounder would never have sexual reproduction taking place. So it's asexual activity that's taking place inside of a test tube, which is also known as in vitro. That's what the important point to remember here is. Otherwise, a lot of people have been confusing people by making statements like, oh, don't worry about genetic engineering, don't worry about a GMO. All we're doing is we're accelerating evolution as how our ancestors used to do breeding. And that's absolutely false because, again, a flounder and a tomato would never have or sexual reproduction ever taking place. A GMO is something that comes in a lab. You know, it's created in a lab, and it's a product of asexual production. Several years ago, our research team, when we saw this ad on the front page of MIT Technology Review, which said "Buy fresh, buy GMOs," I got very intrigued by really wanting to understand genetic engineering of foods. We looked at plants. We wanted to understand what are the fundamental engine within a plant, and one of those fundamental engines is called the C1 metabolic pathway, or C1 metabolism. It's how plants sequester carbon. So when we know this is in plants, it's in fungi, and it's also in bacteria in our gut, for example. So we said, let's take all of these 3 major subsystems and really understand the molecular interactions. Once we put these reactions together, we're able to mathematically understand the different levels of products and byproducts and waste, all these different chemical species. So one of the things we noticed was that for a normal plant, formaldehyde is created, but immediately it's detoxified. So it's created and it goes away, it's created and it goes away, right? So that's a normal cycle. Now what happens when genetic engineering takes place? Well, before we could go there, we needed to understand how that metabolic pathway gets perturbed or gets disturbed. So one of the things we noticed was we looked at another body of literature and we found out that when plants undergo stress, so for example, plants undergo stress all the time, sudden drought, they don't have enough water, they hunker down. And when they hunker down, they start using up their glutathione, which is their antioxidant. By the way, glutathione is one of the most important antioxidants to all life, which means when you create waste products or your body starts quote unquote rusting, glutathione sort of cleans it up. So glutathione is an antioxidant. So when plants are undergoing stress, glutathione comes in, and glutathione levels will start dropping if the drought stays for a long time because you're using up this very powerful antioxidant, and formaldehyde levels will start going up. And then after the drought goes away, they go back to their normal state. So we also discovered this, we modeled it, and we were able to show that to also be true and matched what was in the literature. So the issue is what happens when that kind of genetic engineering took place, and how does it affect the larger C1 metabolic pathways? So in our research, we found 5 different chemicals were altered at different levels. When we took those alterations and we put it back into the original model, lo and behold, what we see is that the plant is actually behaving as though it's stressed out, like in the drought condition. So glutathione levels drop, go to a different level, and formaldehyde actually accumulates. So fundamentally, the plant is in this different mode of operation. It thinks it's under stress, So it's not in its original state, what you would call its original homeostasis. It's in a different state. And it's not homeostasis, technical term, it's called allostasis. The plant is trying to survive, but it's operating in a different mode. If you were to measure glutathione levels, we found that the glutathione levels in a genetically engineered soy plant are 250% lower than the non-genetically engineered plant. When people do their tests of substantial equivalence, as we talked about, they don't measure the difference in glutathione, because that variable will show that the soy, the organic soy and the GMO soy are 250% different. And if that were looked as the criteria, one could argue that plant would never be allowed. The bottom line is this: because there are no objective safety assessment criteria, which means if you're going to compare a genetically engineered plant and a soy plant, no one has established objective criteria. Hey, you have to measure the glutathione levels. If they're not the same, then this plant is not the same. They allow the genetic engineering companies or the GMO food companies like Monsanto to decide their own criteria, which may be irrelevant, like the color of the plant, the amount of water it has, fat, and then they let them do self-reporting. So the bottom line is we have no objective safety assessment standards for genetically engineered foods, period. People have noticed whenever Monsanto or these companies sell their seeds, they were coating them with a substance. It was coated with neonicotinoids. So people have always wondered, why is a genetically engineered seed, if it's so much superior, why is it being coated with something? It's almost to protect it from something. So you've taken a plant, the organic plant, which when you plant the seed of that plant has a pretty good immune system to survive. Forget the pesticide, but the soil organisms in the soil. But if that seed now is inferior, it has less glutathione, which plays a huge effect on the immune system, that plant with less glutathione is a weaker plant. So when you put that seed, into that soil, remember the soil is a very harsh environment. The soil organisms can actually attack it and hurt that seed because it's weaker now. So by coating it with neonicotinoids, which is actually an insecticide, it's actually protecting the plant from the seed, from these natural soil organisms. Now the neonicotinoids, or as I call neonics, actually infuse throughout the entire plant. So consider what we've done. First of all, we throw pesticides on our food. So that's one severe cause of all sorts of diseases, as has been shown. For example, glyphosate, correlation to cancer is pretty high. And then to solve the fact that we're killing the original crop with the weed killer, then they created a Roundup Ready version of that. But then that seed, that organism, is actually not as superior from our data showing because of lowered glutathione levels. So then they coat it with this other thing called the neonicotinoid. The food that you're getting from this has three different things that have been perturbed, right? First of all, the food has levels of glyphosate in it, which is a pesticide. It has lower levels of glutathione, and it also has neonicotinoids in it. There's a growing body of evidence showing that glyphosate, even at low levels, causes cancer. But the more important thing is we don't know the combination of these things, what they cause. Even though you have a little level of this and a little level of this, the synergistic effect of those things can cause all sorts of disturbances in our body. Now you have to understand that the gut microbiome in humans also uses a C1 metabolic pathway. The food that you're eating has less glutathione, but we don't know fully how those pieces of DNA, they can get embedded in, and incorporated into our microbiome. And if you think about it, the microbiome, which is our gut, is one of the key areas that affects all sorts of chemical reactions. So I think that the net of the story here is that you can't make a little itsy-weeny, teeny-weeny change over here and think it's not going to affect this big system. And that's what systems biology is about. And more importantly, nature has taken billions of years if you think about it as an engineer, it's tested things, thrown out things, tested things to hone down that it has found things to be, optimal for certain conditions. So to think we can come change it without understanding all these different changes is naive, at best.